All right. It is my pleasure and honor to introduce tonight's speaker, Dr. Bill Lute. Dr. Lute is the Associate Curator of Fishes at the Natural History Museum of Los Angeles County. He earned his bachelor's degree from the University of Arizona, a master's from the University of Texas at Austin, and a PhD from Louisiana State University. He was a postdoctoral fellow in the Systematic Ichthyology Department at the Smithsonian National Museum of Natural History. His research aims to determine factors that have influenced biodiversity of fishes and determine how patterns of fish biodiversity have changed over time. He also works to conserve and protect marine biodiversity by understanding the impact that we have on our marine environments. To better understand fish diversity, he leverages the variety of data that can be obtained from museum specimens and also actively travels to conduct fieldwork and add specimens to museum collections worldwide. The title of tonight's talk is Using Museum Specimens to Look into the Past and to Inform the Future. Thank you, and please help me welcome Dr. Bill Lute.
All right, well, thank you for that introduction, Julie, and thank you all for being here. This is a great audience, so I'm really excited to speak to you about what I do at the Natural History Museum, kind of what my job is, and what it entails through a couple case studies that I hope are fun for all of us, and to give you all a sense of kind of how museums operate too. So if you haven't been to the Natural History Museum, this is what it would look like if you were to show up. Just a quick show of hands. How many people have actually been to the Natural History Museum? Is that everyone? Has anyone not been to the Natural History Museum? All right. I'm not going to shame you. If you haven't been to the Natural History Museum, I highly encourage you to go. Cabrillo Aquarium is an amazing cultural institution here in Los Angeles, and so is our Natural History Museum. They complement one another. If you love this place, you're going to love the Natural History Museum. If you've already been to the Natural History Museum, I also encourage you to go again because we recently opened this brand new entrance wing called NHM Commons. Really nice new auditorium, new exhibits in there as well, a few welcoming exhibits. The Barbara Carrasco mural is there as well. Great new space, so I highly encourage you all to check it out. Inside our museum, you are obviously going to find a lot of great dioramas and exhibits and displays. The bottom right is our brand new dinosaur that's in the NHM Commons. The name is Natalie. It's a green dinosaur. And these displays and exhibits really not only help inform the public, but they reflect what we have discovered behind the scenes as scientists and the knowledge that we've learned. They reflect our research as well. So a lot of things that you will learn about are things that scientists have discovered using museum specimens. But I'm here to talk about that process, the behind the scenes aspect, kind of what we do and why we do it. And a lot of people, if you've never been behind the scenes of a natural history museum, you might be a little surprised. We are a lot more than just the public side of things. In fact, what's displayed on the public side of things is a very small fraction of what the museum actually holds. And if you were to kind of go through that door and look behind the scenes, what you'll find is a huge variety of collections. In fact, at the Natural History Museum of Los Angeles County, we have over 35 million artifacts and specimens. So quite a bit. And they range everything from cultural and anthropological artifacts to historical artifacts, things especially dealing with history of Los Angeles. And of course, what I'm going to talk about today is more of our biological collections. And we have everything from birds to insects to extinct organisms such as our wonderful dinosaur collections. And really all of these collections, I like to think of them kind of like a library. Each specimen is full of information, just like a book is full of information. We can get these specimens from all over the world. Books come from all over the world. We lend out our specimens to other people to learn from and to do research, just like a library. You can get books from all over the world. So I like to think of them as just a very special type and unique type of library. This is my happy place. This is my collection. I shouldn't claim ownership of it. This is the fish collection at the Natural History Museum of Los Angeles County. It's in one large room on the main site. So this is literally behind the walls as you're walking around. And if you were to look at any one of these aisles, you'll see that we have jars and jars and jars of fishes. In fact, in this room that I work in, there are over 3 million individual fishes. And those are spread out among around 170 to 175,000 jars. So quite a lot of fishes to keep track of. And they're all stored in alcohol. They're very happy where they are, and they'll stay preserved for quite some time. If we're doing our job right, they'll stay good as viable research specimens for over a century. And it's not just the fish in the jar that matters, though, too. Each jar, as you can see, has a label. That label has a lot of associated data with it, where it was collected, who collected it, what depth it was collected at, et cetera, et cetera. And nowadays, there's even more data behind those jar labels in our digital database system, where we can have fresh photos of the field site that it was collected at, even videos of the fish, if we like. So there's a lot of information stored in this one space that we use ourselves for our own research. We send around the world for other people to use. And when I tell people from the public about this space or bring them back, every once in a while I get this kind of quizzical look. And a lot of times this question comes up: why? Why are you doing this? Why is are you collecting? Are you hoarding? What are you doing? And especially some younger people will be like, Bill, I can just Google that. If I want to learn about that fish, I can just Google something. To that I respond, well, one, is your new Google fancy artificial intelligence response. Have you checked to see if it's accurate? And if it is accurate, I'm going to take a claim here that it's probably accurate because of the research that is done at natural history museums or researchers that use specimens from natural history museums. And it's truly kind of hard for some people to get a true grasp of how difficult it is to understand many groups of organisms on our planet, the true scale of how difficult it can be to understand wholly a group. We actually have it pretty easy in fishes compared to some groups like insects that are very diverse. But just to bring it back home to fishes real quick, let's talk about how diverse fishes actually are. So let's consider for a second, just think of all the species of vertebrates on our planet, things with a backbone, things like us. If we look at the distribution of species by vertebrate groups, what you'll find is that fishes make up more than half of all vertebrate species. You can take all the birds, all the amphibians and reptiles, and all the mammals, you can add them all together, and fishes outnumber them. There is currently over 36,000 recognized and accepted species of fishes on our planet. Again, pales in comparison to some groups. One of our marine invertebrate staff members is here today, and marine inverts are far more diverse. But for vertebrates, things like us, fishes are extremely diverse. So there's a lot of fishes out there. In fact, there's other weird statistics you can pull out. Something like 1 out of every 13 vertebrate species is not just a fish, it's a type of catfish. So the fishes are very diverse. And our overall knowledge of many of these species, to be quite frank or quite honest, we don't know a whole lot about many species. If we look at the number of species described each year, too, of new fishes on our planet, what we can find, and if I draw a line here to help orient you, that line is at 365, or roughly 1 new species of fish per day. If we look in the past decade or so, 6 out of the last 10 years, there's been more than 1 new species of fish described per day. So there's a lot of unknown biodiversity out there too. So not only do we not know a whole lot about many species out there, but there's still a lot of species that we haven't even described or discovered. So I'm going to talk about a few case studies here today to give you a sense of how my role as curator of fishes at the Natural History Museum helps us solve some of these difficult things to understand, some of these unknowns that we have about certain species or distributions of fishes. So I'm going to do 3 case studies for you guys today. We're going to talk about a rare species. We're going to talk about regional surveys and new species. And then we're going to talk about change over time and our impact on wildlife. And then we'll wrap it all up at the end and talk about how natural history museums and the data that we discover can help inform the future. All right, so first case study. We're going to talk about a rare species. And this story starts with this beautiful fish right here. It is a fish. It might look like a tarball, but it's really not. This is a large adult female anglerfish. This washed up in about 2021 in Crystal Cove State Park, so not too far from us. This was a very exciting find. It actually made the news. You may remember during that time, lots of newspapers picked it up. If I go to the next slide, you can see this is as we were preparing it. So you can see it's a very beautiful specimen, very pristine, great fish. It made the news probably for the wrong reasons. It made the news because people found this like spooky, scary fish. I don't think she's spooky. I think she's gorgeous and just a beautiful, beautiful specimen. But why it should have made the news is because of its rarity. And I think this point gets lost a little bit. This is number 32. This is the 32nd individual ever of this fish seen. Total. So prior to this, only 31. Everything we know about this species is from 32, well, now a little bit more, and we'll talk about that. But at this point was from 32 individuals. You might be surprised to know, though, that one of those other 31 is here at Cabrillo, so you can see it. But going back to this, everything we know about this species is from 32 individuals. And so when people say, why do we add specimens to collections? Don't we know enough about that fish? Many times, there are true rare fishes out there that, might just wash up. Since that fish, though, been a little bit strange past couple of years. Prior to that fish, there hadn't been one of these species seen in Southern California for 20 years. Since that fish washed up in 2021, though, there's been a total of 5, so 4 more that washed up in Southern California. In fact, there's a 6th that recently washed up. The most recent one washed up in Oregon. But not all of these made it to museums. And in fact, out of those prior 31 that were seen before these all washed up, many of those aren't in museums either. They were either lost or never collected in the first place. And that happened here too. In fact, only the top 3 of these ended up in museums, 2 at my museum and 1 at Scripps Institution of Oceanography. And the bottom 2 individuals were either taken by people not understanding the value of this fish, and I don't mean monetary value. There's no real monetary value in grabbing one of these off the beach, but in terms of the value for knowledge, for learning more about these fishes, or they were taken by scavengers or washed out back to sea. In fact, this bottom left fish was already starting to decompose and not in the best state. So together with my collections manager, Todd Clardy, who's at the Natural History Museum, as well as Ben Frabel at Scripps Institution of Oceanography, we decided to start tallying up all of these fishes, or all these individuals of the species to see if we can gain any insights or learn a little bit more about this fish, seeing as a bunch of these have washed ashore. And well, one question that we do get from the public a lot is, was there some sort of underwater explosion or nuclear leak or something? Why are all these fishes washing up all of a sudden? The answer to that is no. If there was a large perturbation in the deep sea, you would expect our common deep sea fishes to wash ashore, not the rarest of the rare. So we don't really know why these were all washing ashore now, but we do have some thoughts. So we, first of all, just mapped where the known locations of this species were. In California, the black dots are prior to the 2020s, and all of the red dots are the recent collections in California. And we also went through the literature and museum records and just looked at all the known records of this fish, the species, and where it's been found before. And as you can see, there is a positive trend generally for a number of observations of this fish, and the red part of those bars represents individuals that were collected in California. And part of the reason that we think we are seeing more of this species doesn't deal with the fact that they're washing up more frequently. These are all really old, mature females. They're not very small. They're about the maximum sizes this species get. We think this positive trend actually just deals with more people having cameras on them all the time. And so you can actually document them more. That is our working theory. However, we've also learned more about the biology of this fish too, even though there are just a few individuals. So if we go back to our wonderful 2021 specimen, sometimes science works in funny ways. And as Todd, our collections manager, and I were preparing this specimen, Todd joked around and said, "Hey, I wonder if this fish fluoresces." Well, this is an anglerfish. Anglerfishes luminesce. They produce light. In fact, that's what that fancy modified dorsal fin is. It's called an esca. And it produces light to attract prey in the deep sea, the black deep sea, to come up to it so it can eat it. Fluorescence is something totally different. Fluorescence is when one wavelength of light comes in, hits a surface, and is actually reflected a different wavelength of light. So you can have blue light shining on something and then get, a red reflection, basically, or a green reflection. That's fluorescence. So two totally different processes. Todd suggested this, or was joking about this, because we had just purchased some fun new fluorescent flashlights. And, I was definitely like, oh yeah, let's try it. There's no reason to believe that either of us, though, believed that this fish should fluoresce, though. It lives in the deep sea. It lives in a pitch black environment. You receive no external light. Fluorescence needs external light to work. Well, lo and behold, it actually does fluoresce around the esca. There's a lot of rough skin right around the esca of this fish, the lantern of the fish, basically. And it's those rough patches that when you shine a blue light on it, fluoresce these kind of neon green colors. This, we were thinking, is possibly due, or could be due to the fish basically creating its own fluorescence. The fish creates its own light. If that light is the right wavelength of light, that light could be what creates the fluorescence too. So this together could create a more complicated or a more attractive light for attracting prey. This is never described in any deep sea anglerfish before. There is one other example of a deep sea fish that uses a combination of fluorescence and bioluminescence. That's the stoplight loose jaw dragonfish. It uses a blue bioluminescence to create a red fluorescent light that it uses to spot prey. Most fishes in the deep sea can't see red wavelengths because red light never gets to the deep sea, but this one dragonfish can use this kind of red light as a trick to find its prey. This would be a totally different use of a combination of fluorescence and bioluminescence. So even though this was just one individual of a rare species that washed up at one point in time, there's a lot that we can learn, because this now opens the door of, hey, we've never really checked other deep sea fishes for fluorescence. Maybe it is more widespread. So it kind of changes the way that we think or could think about some hypotheses we want to test in the deep sea, all from just one individual. All right, so moving on though, that was rare species. Let's talk about another thing that I do as a curator, and that's doing regional surveys and describing new species. And this is the surveys and the fieldwork aspect. This is the fun part. This is where people think you're Indiana Jones and you go rushing out in the field, you find cool stuff, you grab it, and you say, this belongs in a museum. And you get in fights with people. I've never gotten in a fight with anyone while doing field work, so it's not that exciting. but it is a lot of fun. And so what I'm going to talk to you today is about a regional survey that I was honored to be included on that went out and did an expedition at the Revillagigedos Islands that are far off the Pacific coast of Mexico. So these are very isolated islands. They're volcanic. They are not part of the continental shelf. There are 4 islands in total. Socorro and San Benedicto are the closest. It takes about a day on a boat from Cabo to get to these 2 islands. Roca Partida is kind of in the middle of nowhere, and Clarion is the furthest out. It takes a whole another day to get to Clarion once you're at Socorro. So quite isolated islands, but they're really interesting too. Because they could be, and it's hypothesized that they might be, a stepping stone for species over evolutionary timescales to get from the Central Pacific to the Eastern Pacific. Between those 2 biogeographic regions, there's a lot of nothing, and it's hard to cross that expanse. But these islands may be a stepping stone. These islands are also a UNESCO World Heritage Site. They're a National Park of Mexico. And they're a marine reserve, really popular for scuba diving because they have abundant wildlife. And so there's a lot of people that go every year to go diving, take lots of photos, swim with manta rays, do all the fun stuff. However, there hadn't been a large collecting expedition to these islands for quite some time. And in fact, it had been, since the 60s and 70s that there was a really large expedition that went out here to deposit a lot of material to museums. And so that was kind of the impetus for us going on this expedition. All right, so this is the crew that we all, or that I had the joy of spending a little more than 2 weeks with on a boat. I don't want to point out everyone, that would take too long, but there are a few really key players that were on this trip. This is Carlos Sanchez. He's a professor in La Paz. He was the driving force behind this entire expedition. He reserved the boats. He got all of the permits, which are very difficult to get. He did basically everything in between. This trip would not have happened without Carlos. This man right here in the front, this is Ross Robertson. He's a senior staff scientist at STRI, which is the Smithsonian Tropical Research Institute. He is recently retired, although very active still, and Ross maintains and updates constantly 2 websites. One is a website for fishes of the Caribbean, and one is a website for fishes of the tropical Eastern Pacific. Ross is very particular about what he does, and a lot of this trip was about getting good photos for those websites. They are phenomenal public resources for anyone that's curious about what fish might exist in different places. And because of that, though, Ross's influence made this a really interesting trip. This is not a boat full of scientists. In fact, the scientists are outnumbered by a lot, by amateur dive photographers. So this was a true community science effort to try to get good documentation of every species that we came across while we were out there, not only getting specimens from museums, but getting good in situ live photos. And really the driving force behind the photography team were these 2 people. These are the Estapes, Allison and Carlos. Amazing people, great divers, really good photographers. And so most of this crew is actually underwater photographers. These are the islands. Clarion is the furthest away. That's the one on the top. Roca Partida, again, is kind of in the middle of nowhere, and as the name suggests, it's a parted rock. It's kind of stunning to come across because it really is in the middle of nowhere. Socorro is closer, and San Benedicto might be one of my favorites because, as you can see on the right, it has this big volcanic dome. And in fact, some of the dives there are on these lava fields that are just exquisite. And as you might expect, the seas around the Revillagigedos are just full of life because it is a protected area. You can see everything. You can dive with tunas and other large fishes. There are a lot of sharks out there. Might seem a little spooky, but sharks are actually a very key component of a healthy ecosystem, marine ecosystem. So there should be a lot of sharks out there if it's very healthy. And so it was a great, productive trip. And what did we find, though? What's the whole point? We don't just kind of blindly go out there for no reason. Well, in this regional survey, we have subsequently not only been on this expedition, but since the 2 years since then, we've been scouring through all the records of fishes from the Revillagigedos that have ever been reported, essentially. And I can't take that credit, really. It's Ross Robertson has really driven this project really strongly. And we've confirmed the presence of around 240 species or so using our trip combined with museum specimens. More importantly, though, we have rejected 100 species or so that were supposed to be there. Why is that important? Well, this is a marine protected area. This is some place that is managed, that is protected. To protect a place, you need to know what's actually there. And you might think, how could there be 100 species or so that are supposedly there that turn out not to be there? A lot changes over time in the world of taxonomy and our understanding of fish species limits. What Ross likes to call the detritus of history can be very hard to sort through. And so some of these were total misidentifications. We would go to the shelf, look at a specimen from the 1960s, and whoever identified it did their best. I'm assuming they did their best at the time, but they were just wrong about the ID. Some are due to taxonomic changes. Some are due to a complete lack of vouchers or source. There's just kind of hearsay that the species occurs there. And others we found actually had totally incomplete or incorrect location data. It would say it was like a museum specimen would say it was from the Revillagigedos, and then we would go into the ship logs and it was actually collected somewhere totally different. We haven't been able to confirm about 36 though. So this is still ongoing. However, we are close to submitting this manuscript. And we've gained insight on a number of different species that occur out at the Revillagigedos. Figured out which ones are more kind of always there, always present, which ones are kind of vagrants that just pop in and out. And I don't have time to go into all the things we've learned about the different species, but I'm going to highlight a few. So this species I found to be interesting, what we learned from it, not because of how many we saw there. It was actually, we didn't see any of this fish here. This is a swallow damselfish. We were there in 2022. This species has been reported from the Revillagigedos. It is a damselfish. If you don't know what a damselfish is, you probably actually do. Our local Garibaldi are damselfishes. Clownfishes, so Nemo is a damselfish. This species, though, is in the genus Azurina. They're all kind of pelagic, at the water surface type damselfishes. So they're not associated with anemones. But we didn't see this fish at all, and we were looking for this fish quite extensively. However, some of our colleagues went back in 2023 and noticed a few small juveniles of this fish at Roca Partida, and then they went back in 2024 and found a number of young adults at Roca Partida, suggesting that this fish goes through kind of boom and bust cycles at the islands. This is important because this fish is closely related to the Galapagos damselfish of the same genus. The fish on the right, though, hasn't been seen since the 1980s. There was a really warm El Nino event that hit the Galapagos in the mid-1980s, and this fish is now presumed extinct. So it seems that both of these species tended to go through these large population fluctuations. And so it is interesting that we didn't come across this fish at all. However, there is probably more hope for the swallow damselfish because it does occur in places other than the Revillagigedos. So it occurs in a couple other offshore locations and has been actually recorded at out at the Channel Islands periodically, not there all the time. Another fish that we came across, though, that I think is really fun, we found on the very last day of the dive. So we had been, or the last day of the trip, we had been diving for over 2 weeks, doing a lot of dives, working really hard. Last day came, and everyone wanted to have a fun dive. So we didn't want to collect anything. We wanted to just get in the water. Everyone took their cameras, just wanted to see some fun stuff, except one person. Except for Carlos Sanchez, the guy who organized the whole trip. He took a little net, just in case. And after the dive, all of us were talking about, oh, we saw this big shark, we saw this, and what'd you guys see, what'd you get photos of? And Carlos brought out these two fishes. It was really interesting, these little red and white fishes with these kind of black dots on them with yellow margins. They're only about an inch or so long. They're not too big. And immediately, everyone was really interested in these. And it's because these aren't fishes that are reported from the Revillagidos. They actually look fairly similar to a species of wrasse that is found on Cocos Island, which is another offshore island that is in the territory of Costa Rica. But nothing like this has been reported for the Revillagidos. And so we really weren't sure what we were looking at, whether or not it was the same species that occurs in Cocos Island or something totally different. And so we had to go search for more, though. And we had to go search for more because this is a wrasse. Wrasses are sequential hermaphrodites. They actually start life as females, and then they get bigger, and they become males, and they change phenotypically. They change how they look. Great example on our coast is California sheephead. It looks different when it's younger. It looks different as an adult. These are all small individuals. You know, we have initial phase females here on the left and a juvenile on the right. So we were in search of the adult male. Well, what do you look for if you don't know what you're really looking for? And so we had an idea to try to look for terminal phase male wrasse that looks something like the male wrasse from Cocos Island. And we only had 2 more dives to do this on the whole trip because it was the last day. So on the next dive, we did manage to find 2 different males, but they were very hard to get photos of. Too far for the photographers to get good strobe flashes at. And so the colors are distorted. However, we did notice it looks similar to the male of the species that occurs on Cocos Island, but it's different. The patterning of it is different. But we still needed to collect a specimen to see and try to truly identify what it was. And so on the very last dive, we did manage to collect one. I put in this picture of us working underwater just to really highlight the type of habitat that we were in too. So we're about 60 feet depth of water, and it's really just kind of rocky rubble habitat. But we got the fish, and this is what these 2 individuals look like. We have a terminal phase male and an initial phase female. And this is when we started to really do comparative work, comparing these specimens with specimens that are housed in our museum and other museums to see if there's a difference between some of these different forms that occur at these different offshore islands. But before doing that, we had to have some more fun with fluorescence because I brought the fluorescent flashlights onto the trip. And so, but it's really cool. So this is the same species, just male and female of the same species. You would think maybe they fluoresce the same way. They don't. Under blue light, the female will glow this really bright red color, whereas the male almost doesn't fluoresce at all. You really don't see much except some of the fin rays of the dorsal and anal, and the very tip of the caudal fin will fluoresce. But completely different fluorescent patterns for the same species. All right, well, then we got back to shore. Then we did the actual comparative work. We also sequenced genetic data as well. And it turns out this was a completely new species unknown to science. So this is the Revillagidos. This is a very common dive site. There are hundreds of divers every single year here photographing fishes. And yet we still found an undescribed species. And one that was fairly obvious that it was something that shouldn't be there. And this just points out that there's a lot more exploring and species to discover on our planet than a lot of people think. A lot of people think, oh, the age of exploration is over except for going into space. Not true at all. There's a lot more places to explore and a lot of new species out there to find. All right, so that's the end of the second case study. Both of these case studies are kind of what I like to call the what's that fish part of my job, which is to just figure out what a fish is. There's many other types of studies that you can use museums for, though. And I like to think that they kind of fall on a spectrum of empirical studies to more applied science. My lab does both of these. On the empirical side, we've done a lot of work trying to figure out, using genomic methods, how fishes are related to one another. This is a very difficult problem. And so today, this was a publication that was led by a few scientists at Yale, but was a true large international collaboration. And to date, it is the largest and most rich data set of how fishes are related to one another. This type of stuff really gets me going and keeps me up at night. I do understand it might not keep all Angelinos up at night. And so for the last part of the talk, I'm going to go more the applied route of things, or the applied science of things. And we're going to talk about change over time and our impact on wildlife, specifically with Southern California fishes. All right, so for applied science, I like to think, let's start here. I don't know if Bill and Ted is your favorite time machine. Maybe you're a Doctor Who fan, maybe you like Back to the Future. My favorite time machine is a natural history museum. Bet you didn't see that coming. It is the only actual working time machine on our planet. I can go back to a stream in 1930 on May 6th. I can pick up the fish and hold it, the fish that lived in that stream. I can also grab another fish that was collected at the same time and get a sense of the community of fishes that existed at that point in time and location, maybe 50 years ago, maybe 100 years ago. Only working natural time machine on our planet, according to me. So what we're going to do in this last bit, though, is quickly travel back in time and search for microplastics. And so just a quick background, if you don't think about plastics a lot, a plastic is just a synthetic material made from a wide range of organic polymers, such as polyethylene, PVC, nylon, etc., that can be molded into shape while soft and then set a rigid or a slightly elastic form. So that's just a really fancy way of saying these are complicated chemical compounds and polymers that we use to shape and make a lot of cheap stuff. Plastics, though, let's do a really brief history. Started really not that long ago. 1907 was the first commercially really produced in mass plastics with bakelite. So really just slightly over 100 years. Bakelite, you can still find these in, antique stores. It has kind of this dull brownish kind of facade to it. But very quickly things changed from bakelite. So from 1907, just 20 years later, we start to see things that you will recognize. These are plastics that are made mostly from oil or oil derivatives. And the first really big one was PVC. Anyone who's done any plumbing, you probably have seen PVC. Shortly after that, we have polystyrene. You're going to know it as styrofoam. Shortly after that, we have polyamide, which you're probably going to know as nylon. So just by 1935, we all of a sudden have plastic clothing. In fact, all of us probably have plastic in our clothing that we're wearing right now. Quickly after that, though, something really important happens in the history of plastics. And it's World War II. And it's specifically the American involvement in World War II that is critical for the history of plastics. Why is this? It's because plastics were made for the war effort. Everything from clips on the helmets of soldiers to actually airplane windows started going from glass to a type of plastic to things that the soldiers needed to use, combs and other things. Plastics also were used to substitute domestic products. You know, there's a lot of things that were in short supply domestically while the war effort was going on. So instead of having natural rubber tires on your car that you were driving around in the US while the war was going on, you would have a synthetic rubber tire that had plastics in it as well. And during this, there was also a shift in manufacturing to meet this demand. And in fact, between 1941 and 1945, there was a fourfold increase in plastic production in the US. Well, what happens after World War II? All of this industry has already shifted into making lots of plastic. They don't want to go back. They've already invested a lot of money into the production of plastic. And so after World War II, you start to have this age of consumerism, cheap things, disposable things, make your life more convenient. And from this point on, including, you could argue to right now, we've lived in this stage of plastic production and consumption. However, in the 1960s and 70s, we also started to notice maybe some bad side of plastic. This is when there were the first records of seabirds that were consuming plastic, dying from it. First publications of marine plastic pollution was during this time period. And this is also the time period of the more contemporary environmental movement. You know, think Silent Spring and other things that were going on during this time. If we look at plastic as a whole now, across all of history, 8,300 million metric tons have been produced. Some of that's still in use. Oh, and this is from 2017, so this is outdated now. Some of that's still in use, but out of that, 6,300 million metric tons have gone to waste. A little sliver of that's been recycled. However, we could also debate whether or not that's truly recycled. A bigger chunk of that has just been incinerated and burned, but the vast majority of that is either in landfills or out in the environment. And what happens when plastic is out in the environment? It's exposed to sunlight, it degrades, it becomes very brittle. It breaks apart and breaks apart, breaks apart and breaks apart, breaks apart. It gets smaller and smaller and smaller and smaller into these smaller particles that we call microplastics, although on this screen they are quite large. So a microplastic is any particle, according to NOAA, that is 5 millimeters or smaller. 5 millimeters is about the size of a pencil eraser. So anything smaller than that is considered a microplastic based on the US definition. But there's a lot going on with microplastics too. They're pervasive, they're everywhere, from the tallest mountain peaks to the deepest ocean trenches. They're diverse in terms of shape, in terms of chemical composition, and they're really an underappreciated component of pollution. However, they have been making the news a lot more recently. And what I wonder about microplastics, though, is when did they first start appearing in the wild? Are animals eating them, and is this consumption increasing? And what trends are we seeing across time? This is something that we can actually answer using our museum time machine. And so I'm going to very quickly show our kind of experimental setup. What we decided to do is look at multiple different species with different diets, see if diet has an impact on whether or not you consume plastic. Right now, what I'm going to show you is only for Southern California. So the line on that map is Point Conception. Anything south of that we considered to be Southern California that we could collect samples from. We wanted to see what type of plastics there are out there. And we wanted to see how this changed over time. And we chose from 1940 to the present to kind of correspond with that increase in plastic production in the US. And so the different species that we're looking at this in, we're looking at it in the northern anchovy, an opali. Opali is an herbivore, it eats algae. We're looking at shiner perch, which is kind of a more generalist fish, eats invertebrates, and California halibut, something that eats other fishes. We're also not only looking in shallow water fishes, we have chosen two fishes that you might think of more as deep sea, the northern lampfish, which is a type of myctophid or lanternfish, and the Pacific viperfish, very toothy type fish. This work is largely driven by Jessica Flores, who is here tonight, and Jasmine Gallardo. They're two of my lab members. They're fantastic at what they do, and they've really driven this project forward. This is not going to do them service for me to say that what the protocol is. No, it's definitely more complicated than this, but conceptually, it's really easy to understand. You take a fish, you cut out its gut, you digest it in a strong base, and then you filter it onto a glass filter and look at what you got. A lot more complicated than that. Sorry, Jessica. But that's conceptually how we do this work. We can look at these filters and we can see if there's plastic particles on them. You can see on the right, there's that red fiber, and we can do visual inspection of these filters. Visual inspection, though, is actually quite biased. One, we're very bad at detecting really small plastics, and we're trying to get to the nano kind of stage. But it's also hard when something's kind of clear. Is that a piece of cling wrap? Is that a shell of an animal? Kind of hard to tell. Even actually can be hard to tell, especially for the really small stuff, even when you try to stain these with kind of fluorescent dyes. So what we use instead is a Raman spectrometer. And this is a really fancy laser machine that just shoots a laser at a sample and kind of sniffs the smoke that comes up to tell you what the chemical composition is that can tell us general kind of plastic types that we find. All right, before I tell you the results, I will say this is not a complete study. We are actually quite early on in this study. So I'm not going to talk about trends over time because I don't want to be misleading if our future samples that we're finding change our understanding. However, I am very comfortable talking about parts of this study because if a plastic's there, plastic's there. That's not going to change with future samples added on. But we are early on in this study. And so far, we've only started to really examine 4 of these species. The myctovid is the one that we have sampled completely. Everything else, we still need to do more species. But we have deep sea and shallow water fishes represented. We have different diet preferences represented as well. And what have we found? We find plastics as early as 1956. So far, nothing earlier, but that is the earliest that we find them, and very consistently from the 60s onwards. Plastics have been found in every single species that we've looked at. Doesn't matter what you eat. Doesn't matter if you're deep sea or shallow. We don't find evidence of bioaccumulation. That is the one thing that I did bring in here. I'm going to put an asterisk on. This could change. However, we don't see predators having more plastics than things that are lower on the food chain. And we found, of course, multiple plastic types, everything from polyethylene to nylon fibers to polypropylene, et cetera. Interestingly, though, on the right, this is a Raman map, and the colored things are plastic particles. Interestingly, though, plastics aren't in every fish. Even in some modern samples, we can do a fish from the last decade and find it completely empty. However, some fishes, like this one that is represented on the right, are full of plastics. So it varies a lot, actually, between individuals. So why does this matter? Why should you care? Well, it's a very pervasive problem. Marine organisms are eating plastics instead of the food they should be eating. This can lead to less nutrients in the fish. Their stomachs can get full of plastic. Or it can be really important at certain critical life stages too. Think the larval stage of a fish. They're very susceptible at this time point. They're supposed to be eating little things in the water column. They're confusing them with plastic. It can be very detrimental to that life stage. The chemicals used in plastic production, many of them proprietary blends by these companies, have impacts on reproductive, nervous, and endocrine systems. And this is not just a fish problem. These plastics are getting everywhere to deep sea marine sediments. It's really an ecosystem-wide problem. Not only that, but I'm sure you guys have heard the saying, you are what you eat. Well, if we look at where plastics have been found in humans, whether or not you got it by eating a fish or just by living on our planet right now, plastics have been found in the brain, skin, lungs and breast milk, your blood, liver, stomach and intestines, even testes and placenta. Plastics have been found quite widely in the human body and are likely having adverse effects on us as well. All right, so let's lift up a little bit from that little negative slide. But if we go back to the collection, where my happy place is, hopefully I want to bring it back home and think about how natural history museums can help inform the future. Now, as the title of the talk, kind of want to finish with rounding up some of these examples and see if we can relate back to that. You can't protect what you don't know. You need to know what species exist where and what species even exist. You also can't document a problem in our litigious society if you don't have physical evidence. It's really hard to prove something without some hard, concrete evidence. And you can't go back in time. Unfortunately, Bill and Ted doesn't exist in real life, or whatever your favorite time machine is. We can't go back in time to gather more data. But natural history museums, we do document and describe wildlife on our planet. I haven't talked about it, but we hold what are called type specimens. These are the individuals used actually in the description of a species and are the ultimate authority on what a species is. We house vouchered material that can be referenced and rechecked over and over again. And we work as a time machine. Again, we're the only working time machine. We can go back in time. We can see if there's trends, if there's anything that happens and changes over time. And I haven't talked about it, but we can also, instead of going across time, we can go across geographic space and look for spatial patterns too. And we're a repository and a resource for the wider research community and the public. So I haven't talked about it much, but we do a lot in terms of public education, just like y'all do here at Cabrillo as well. And we also train future scientists too. So I hope the next time, or maybe the first time, that you go to the Natural History Museum, you think more about just what you see in the dioramas and think about what's behind the scenes. However, I do want to end this on kind of a call for action slide. And that is, we are living in kind of a very bizarre time. And the support for science or the value of science and even the acceptance or accuracy of science is being doubted. It's being called into question, even actively combated. And it truly takes a whole community effort to support the type of research that I've shown today. Ticket sales don't cover the research. That's not the point of the ticket sales. Really, to do this research, you need federal support, all the way down to state and local county support, to private donations as well. And so I just would like to call for action, because we are living in weird times, that if you value science and service that natural history museums provide, please support your local institutions, including places like Cabrillo as well, that are fantastic educational resources too. With that, I couldn't do any of this without the help of a lot of people and these funding agencies too, including federal sources. And I want to thank you for your time.
Thank you, Bill. What a wonderful talk. I would like to ask my staff to turn on the mic so the folks at home can hear the questions, and just reminding all of you at home, once those turn green, it is live. Yes, so now it's live. Everyone at home can hear you. So just watch what you say if you don't want anyone at home hearing you. But I will turn it over to Bill to ask some questions, answer some questions.
Anyone have any questions? Yeah.
What do you store the fish in? Is it formaldehyde?
We initially, so the question is, what do we store the fish in and whether or not it's formaldehyde? We initially fix the fish in formaldehyde, then we rinse it off with several washes of water, and then we store them long-term in ethanol.
So the follow-up, do you test the formaldehyde or whatever you do for plastics at all before you?
So that is a good question, and we can't go back to test the formaldehyde that someone used
nowadays
50 years ago. No, we don't test the formaldehyde, and we typically also don't inject the formaldehyde though into the digestive tract of the fish. That can happen, but that's not something that we've been looking into, because we can't standardize that across time. Could that be a source of extra plastic? Sure. In fact, Jessica, who is the person that does a lot of this lab work, will tell you the hardest part about this is just keeping out contamination. That is by far the hardest part of microplastic work, because microplastics are everywhere. I mean, everywhere. In fact, we realized that there's a whole brand new pack of filters that we opened up. Before we switched to only using the glass filters, we were using some 100% cotton filters that we decided to check before even doing some of the work, and found that there were microplastics embedded in those filters, that we couldn't use them, essentially. So, yeah.
How do you decide when to use and how much to use of destructive testing? I mean, I understand you've got all these samples and you keep them for years, but as people do this testing, are the samples going to get smaller and disappear?
This is a fantastic question, so thank you for asking. And it is, how do we decide when to allow people to destructively sample a specimen? Obviously, these specimens are priceless. You can't go back in time to collect more. And so we have to take a very careful approach. And the reason that we chose the species that we did is because of the number of those specimens that we have. There are a lot of other species that we could probably pick from, but we didn't have quite the numbers that we wanted. And we don't want to damage every specimen that we have of a species. So this is also why it's important for us. It's not really, when we go out collecting, we don't collect just one of a species and say, that's it, that's enough, I got it, I got a representative of that one. We try to collect multiple individuals, so there are redundant samples that can be used for destructive sampling. But a great question. Yeah.
So when I first think about plastic, I'm only thinking about plastic bags causing an issue, but microplastic is something I just found out today. if it's accumulating in deep sea, would that be one of the reasons why maybe the football fish and all these deep sea fishes are floating onto shore? And also, since we're having a problem with the deep sea microplastic, I'm assuming space is probably have the same thing with plastics in space.
Yeah, this is also a good question of whether or not microplastics are causing some of these deep sea animals to die and wash ashore. Those fishes, the anglerfishes, what's really interesting about all of those that have washed up have been completely empty stomachs, like nothing in their stomachs at all. Now, do they have microplastics? Probably. The size of the fish means that they've been around for quite a number of years, and so they've likely accumulated microplastics. It's hard to say though whether or not that is what caused them to die.
So when you get the specimen, do you check, do you examine for the microplastic?
A specimen that rare, no.
No.
We try not to damage the specimen, because it's such a rare species, we really try to destructively sample it. Because to really also determine microplastic, you need a good sample size of fishes, and we just will, I know that we won't get that of those extremely rare species. Yeah,
The anglerfish, you've only found females?
so this species, you might know for anglerfish, maybe you don't know. Anglerfish are really cool for a number of reasons. The large individuals are females, males of many anglerfishes, and there's over 160 species of anglerfish. Males, for many species, are parasitic. They're very tiny, they latch onto a female, they actually fuse to the female, and they take a free ride for the rest of their life. Whether or not you think that sounds pretty nice, probably depends on, a number of things, but we did not find any males of this species. In fact, no males have ever been reported of this species. Hemantolophidae, where the family of anglerfishes that this rare species comes from, they're actually not parasitic males. So parasitic males is not a uniform thing across deep sea anglerfishes. And so we check them to see if there's any parasites on them, parasites for Julie, but we haven't seen any males. In the back.
How do you know that the males are not parasitic if you haven't, like, can you definitively say that the males are not parasitic, or are you just assuming it because you have not come across it?
So that's a good question. And I guess we are inferring that they're not parasitic for this species. However, this is not the only species in the family Hemantolophidae. And for those other species, there have been males reported. And so they have a different morphology than when they would fuse onto a female. So we assume that it's uniform for the family Hemantolophidae, that they're not parasitic. Yeah.
You still have a flashlight.
Do we have the fluorescent flashlight? Yeah, I actually have more now. Yeah.
Determine why the male versus the female wrasse fluoresce so differently? Was it hormonal, or is it chemical, or just gender related?
That's a really good question that I don't have a good answer for. It is something to think about though.
What's the question?
Why the new species of wrasse that I showed, why the males and females fluoresce differently. But I don't have an answer for you, unfortunately. Yeah, up in front. What? A laser? Yeah, so to identify what type of plastic, because there's a bunch of different types of plastics. And to figure out which type we're looking at, because it can be really hard when they're small fragments, we shoot a laser at it. And when we shoot a laser at it, some of the chemicals in that plastic kind of come out, and the machine can actually detect that, so we can figure out what it is. Yeah.
Wouldn't that be dangerous?
Lasers can be dangerous, yeah, but it is protected. The machine protects us, so we're not like staring right at the laser, because that would probably be bad for our eyes. Someone probably could, but I'm glad they don't. Yeah.
So with biocomposable plastic, that still creates microplastic, correct? Because this misnomer we shouldn't be using microplastic.
Yeah, so the question deals with compostable plastics, which you might have seen. Compostable plastics will compost in an industrial compost situation. You can't put those in your backyard compost. They're not going to compost and break down that way. So yes, they can create microplastic particles if they're not disposed of properly.
So it's just the industry way of trying to tell the consumer it's safe enough to use, but it's not.
If you dispose of it properly, and you know the green bin that goes to the big industrial composting, it will compost. So whether or not you think it's industry kind of trying to make itself look better or not, that's going to be up to you. But yeah.
I have a question, I wonder if you can use other method that is less destructive to test if there are any microplastic in the sample.
So if there are other ways to detect microplastics that aren't destructive, is your question? Do you have suggestions?
Micro CT, MRI.
So we do have a micro CT machine. It's another toy that I didn't show today, but it is one of my favorite things to play with. If we did CT, our CT scanner only gets down to about 3 micron, and we can get finer resolution on the ramen. It's also hard to distinguish though between like a shell in a fish's stomach as their last meal, or a piece of plastic using a CT scanner, and we can't tell what type of plastic it is. So could there be other ways to detect it? It is possible. These are also really hard to detect. And it is unfortunate that it is destructive sampling. It's not the only destructive sampling that is done on museum specimens, either by us or other scientists. We approve destructive sampling because it is a way that we learn a lot about the life on our planet. Yes.
For your 1956 first detected in your study, do you have an estimate for how long it took the plastic to get to that state? And then also, do you work in a clean room environment?
We have created as clean a room as we can make at the museum. We are dealing with a building that was opened in 1913. But we do all of our work under fume hoods, or actually its own ductless fume hood system that is not connected to any other ducts in the museum. So we do try to make absolutely as clean of an environment as possible because again, contamination is a serious issue. Regarding your questions of how long it takes a plastic to break down to become a microplastic, it depends, I think, it would depend on the source of the plastic because microplastics can also come straight from the factory. In fact, a lot of these plastic manufacturers make what's called nurdles. And these are tiny pieces of plastic that other manufacturers will use to melt down and press into forms. And so if maybe a container of nurdles gets turned over in the ocean or something, that can be direct microplastic release. So it's really hard to say how long it takes for that piece, like how long did it take for whatever plastic to break down to end up in that 1956 sample? Was that plastic that was created in 1940, 1950? It's really difficult to say. Yeah.
Have you encountered any oarfish specimens?
We have oarfish specimens at the museum. You should come by because there's actually one on display. And we have several other oarfish behind the scenes as well. They're really cool fish.
I have a question. It goes with the question you asked about the destructive sampling. When you're choosing the individuals that you're going to dissect or dissolve, are you looking at a lot and see how many are in a lot? Maybe explain to everybody what that is.
So the way that we store specimens in a fish collection is each jar we actually call a lot. It doesn't mean there's a lot of fishes in it. There can be a lot that's a single fish. Each jar is called a lot. And all a lot is all the individuals of a single species that were collected at the same time. So if we go sampling and we get like 3 bluefish and 2 redfish, those 3 bluefish would go into one lot and the 2 redfish would go into one lot. And you're asking is, do we would we destructively sample a lot of like a single individual, or are we looking for lots that have multiple? Ideally, we do look for redundant samples, so lots that have multiple individuals. We also have to constrain our sampling based on size of the fish and location of the fish. All of these fishes were not collected for the purpose of this study. That's just kind of the reality of this type of study. And so we have to be very careful and choose. We have to balance these things, where the fish comes from, what decade it's in, how many other specimens we have in it, how many lots we have it. It's a delicate juggling act, so it can be difficult. But yes, ideally, we take a fish from a multi-individual lot.
Any other questions? Let's maybe take one more.
Yeah.
Well, you could have two questions, it's fine.
What's the question?
Do about the plastic?
Hardest question for last. What do we do about this microplastic problem? Great question. Totally loaded question. Very hard to answer that question. Obviously, there are definitely things that you can personally do. Reduce your plastic use, reuse things, don't buy plastics. You can put a filter on your dishwasher to stop plastic fibers from entering the water source. There's a lot of things that you can do. The scope of the problem, though, is ginormous. I mean, the scope of microplastic and plastic pollution in general is absolutely humongous. And it is kind of hard. There are things that you can do yourself, but it is kind of hard to see a large solution to this without large legislative action, really large scale change. And so you can be a very positive advocate and reach out and let your representatives know that you are really concerned about this. This is a serious problem because it is a very large problem.
Okay, thank you, everybody. If you would join me in a round of applause for Dr. Lute, what a wonderful talk.